Now let's talk about the genesis of RBCs, that how the RBCs come to form. Now, whenever we talk about the formation of red blood cells, we have to talk in its stages. Now, the first stage or the first phase is the one where we have both committed and non-committed stem cells. The first stem cell, which is usually found in abundance in the bone marrow, is the pluripotent hematopoietic stem cell or the PHSC. Now, this pluripotent hematopoietic stem cell can divide itself into multitudes of further progenitors. It can divide itself into either a lymphoid stem cell. Now, this lymphoid stem cell can further give rise to both lymphocytes and the NK cells or the natural killer cells, or this stem cell can give rise to a multipotential progenitor. Now, this multipotential progenitor is known as the CFU-GM or the colony forming unit, granulocyte and monocyte. Now, again, this is again a non-committed progenitor. It can further give rise to either CFU-GEMM, that is the colony forming unit, granulocyte, eosinophils, megakaryocytes and macrophages, or which then form the platelets and other types of the white blood cells, that is the monocytes, basophils, neutrophils, or it can give rise to another type of progenitor. Now, this progenitor is known as the CFUE and CFUB, that is the colony forming unit erythroid and colony forming unit basophilic erythroid. Now, both of these CFUE and CFUB do not have the capability to transfer into another type of a progenitor, because at this stage the stem cell has differentiated enough to be called a committed progenitor. Now, at this stage, the progenitor cells do not and cannot give rise to another type of progenitor. So, if we were to incubate, let's say, a stem cell in a laboratory condition, it can give rise to both lymphoid stem cells or the CFU-GM stem cells, because both of these lines can be found after the division of the stem cells. But if you incubate the CFU-E and B, the committed progenitors in laboratory conditions, only one cell line will be formed. So if you incubate CFU-E, you will only see the amount of the erythroid cells being formed. And if you incubate only the lymphoid stem cells, only the lymphoid cells will be formed. So after the formation of a committed progenitor has happened from the non-committed one, which has differentiated itself enough from the other colony forming units, it gives rise to the second phase. Now, this second phase is the division and differentiation of the erythroid progenitors. Now, these erythroid progenitors, like I said, only follow one cell line. In this case, as we are learning erythropoiesis, they will only give rise to the progenitors of the RBCs. Now, these ones include the now it is worth noting that all of these cell lines are the ones which have some kind of effect on the formation and the production of erythropoietin, or they are responsive to them. Now we will learn about erythropoietin in a further down the slides. Now these committed or erythroid progenitors are the first one being the proerythroblast, which we learned in another bite, which gives rise to the basophilic erythroblast, which gives rise to the polychromatic erythroblast, which gives rise to the orthochromatic erythroblast. Now, orthochromatic erythroblast gives rise to the last phase. Now, all of these progenitors, from the proerythroblast to the orthochromatic erythroblast, are known as the erythroid progenitors because they can only go in one direction and form only one cell line. And we have discussed all of these progenitors in much detail in the stages of the erythropoiesis. Now, at the stage or the phase 3, that is the terminal maturation, what happens is we have the formation of the reticulocytes. Now, reticulocytes are the end stage of the phase 2 and the start of the phase 3 because at this stage the progenitors have differentiated themselves enough to resemble RBCs, but they have not matured enough. Once a reticulocyte is formed, it is then released into the bloodstream. Now, it is released into the bloodstream via a special process known as the diapetesis, and I have already explained what diapetesis is. Now, once this reticulocyte enters the bloodstream, it forms the mature RBCs. Now, this mature RBC or the erythrocyte happens because the reticulocytes are known as reticulocytes because they contain the reticulum. And when they release the reticulum outside of their cells through either exocytosis or simply releases them, or they can phagocytosis, all of these three things can happen. It will then further mature into a normal enucleated RBC. So the end stage is the terminal maturation and it takes place in the blood. While the first two phases took place in the bone marrow or the site of the erythropoiesis, the last stage, the maturation stage, takes place in the blood. And if you do not have enough maturation factors which are needed for the maturation of reticulocyte into a matured erythrocyte, the proper maturation doesn't happen. And depending upon the deficiency of the maturation factor, variety of effects can happen. For example, if we have low amounts of vitamin B12, the shape of the RBCs cannot be maintained. Instead of a biconcave, they turn into a bag-like entity known as the macrocytes.